Hello viewers, welcome back to my channel. Today we are diving into one of the most essential building blocks of any SDTM submission, the demographic domains or the DM. This domain forms the foundation of subject level data. It's the first place reviewers and statisticians go when they are trying to understand who participated in a clinical trial. By the end of this video, you will know exactly what the DM domain is, why it matters, what variables it includes, how it links with other domains, and you will be ready to tackle real world examples and interview questions with confidence. So let's get started. The demographic domain contains the core subject level data collected during the trial, including each subject's unique identifier, sex, race, age, country, and other baseline characteristics. This domain is the part of the special purpose domain in SDTM, which are essential for identifying subjects and linking information across other domains. The structure of this domain is one record per subject. The topic variable is not the subject ID, but the subject ID. The required variable in this domain are the study ID, domain, subject ID, site ID, sex, and country. The expected variables are the date variables mainly, which are the reference start date, reference end date, reference study exposure start date, reference exposure end date, the informed consent date, the participation end date, death flag, age unit, race, arm code, actual arm code, arm not reported, and arm not reported description. The permissible variables are again the two of the date variables which are regarding the start and end date for the uh, inducing agent if used in the study, then the investigator ID, the investigator name, the birth date, ethnicity, DMDTC, and DMDY. The DM domain is crucial because it uniquely identifies each subject. It provides subject level attributes required for analysis. It also links with almost every other domain using the uSubject ID. It forms the basis of subject disposition, demographic subgroup analysis, and the study population summaries. Another point to note here is that all the date variables will be using the ISO 8601 format for its values. And for the country variable, we are using the ISO 31661 alpha 3 format. Now let's understand the uSubject ID, subject ID, and site ID in SDTM. Every subject in a clinical trial is assigned a subject ID, which stands for subject identifier. This ID is unique within a study. It's how we recognize a subject in that particular trial. But if a subject participates in multiple studies, maybe in different phases or follow-up trials, then how we will identify that subject across the trials. That's where uSubject ID comes in. The uSubject ID stands for the unique subject identifier, and it ensures the subject can be uniquely identified across all studies, database, and submissions involving the same product. So while subject ID may represent or repeat across different studies, uSubject ID must be globally unique. Let's understand this with an example. Here I'm showing you dummy data for two of the subjects, say for trial 001. In this dummy data, you can see that the subject ID is the 001 and 002, and the uSubject ID is the concatenation of a code which is given for the study ID and the subject ID, which is P001 and 001 as the subject ID. So we are having two records for these two subjects in DM. Now let's say if the first subject, which is the subject number 001, having this uSubject ID in the trial 001 as P001 0001 as a uSubject, so uSubject ID also enrolls in another trial, say trial 002. Then what should be the U subject ID and the subject ID for this subject in the second trial it enrolls to, and how we will identify that this subject has enrolled in more than one trial. So here if you see the dummy data for the trial 002, you will notice that we have kept the U subject ID same as the one which was assigned to this subject in the first trial, that is P001001. So it is an important factor here that the U subject ID should be globally unique if the subject is enrolling or taking part in more than one study at a time. Now let's talk about a more complex scenario. What happens if a subject changes site during their trial? Maybe they move from site 1 to site 5 due to relocation or logistics. As a sponsor, you need to decide how to manage the site ID, subject ID, and use subject ID. Importantly, only one record should appear in the DM domain per subject, even if they change site. If you need to capture any additional details like the fact that the subject transfer between sites, you can use the supplemental DM dataset. This allows you to maintain a clean DM domain while still documenting important events like site transfer. Let's see this concept with an example here. Here you can see that we have a data for two subjects again, and if you see the second table which I am showing you, which is the sub DM, here this for the subject 001, that subject who was initially assigned to the site 001 has changed the site again later on during the study to site 005. How we can identify that? You can see in the supplemental dataset, the ID var and ID var val have the information of the subject ID for which these records are created, and the QNAM and QLabel gives the description of what information they are capturing. The QL is giving the answer to the question you have raised or the information you are capturing for QNAM. So you can see here that from subDM, the subject ID 001 has a record for site transfer to 005. So that means the subject 001 who was initially assigned to 001 site ID in your DM domain has a site transfer to site 005. Another thing to note is that here in subcall we are also capturing the information of the date of site transfer as another record for the subcall domain. But the most important thing to note here is that in DM we are maintaining only one record per subject, and the site ID is capturing the initially assigned site ID. This setup makes the U subject ID consistent across all records and dataset. It helps to keep the DM clean with single entry per subject and captures the site change details in sub DM. So here we are not losing any information and we are capturing all the information which is captured at the site trial. Let's talk about how multiple race responses should be handled in the SDTM demographic domains and supplemental qualified dataset, that is the sub-DM. When subject reports more than one race, the race variable in DM should be set to multiple. This signals that multiple races were collected. However, DM only stores a single race value, so the additional race information needs to be included in the sub-DM, which allows for multiple rows per subject and can capture all extra details. It is important that both DM and sub-DM use control terminology for race variables. This consistency allows for clean and accurate summaries or analysis, whether the race data is found in the main DM dataset or in the supplemental rows. If a subject refuses to provide race information or it is unknown, the race variable should be coded as unknown. All of this ensures that race data is accurately represented in a way that aligns with FDA and CDISC standards, making the data easier to interpret and summarize. Let's discuss about the key rules for the population of ARM code, ARM, actual ARM code, and the ARM NRS variable. The first one is that when the ARM code is null, then ARM must be null and the ARM NRS variable must be populated. This is a scenario where you can see from the example of the data I'm showing you below that the ARM code is null for two scenarios where it can be for the screen failure or the subject was not assigned. So in this scenario, since the subject was not assigned to any ARM or it was a screen failure subject, in that scenario, the ARM code and ARM code will be kept as null. And the reason you can say why these are null are captured in the ARM NRS variable. The same is the case with the ACT-ARM CD, that if ACT-ARM CD is null, then the ACT-ARM must be null and the ARM NRS variable must be populated. This also comes in the scenario where the subject was either screen failure or not assigned. But there is another scenario for which this rule is true is that the subject was assigned to a particular arm, but the subject did not receive any treatment. So in that scenario also, the actual arm and actual arm code can be null, and the description of this is captured, the coded value you can say the is captured in the ARM NRS, and the description will be captured in the ACT ARMUD. And the third scenario is that when both ARM CD and ACT ARM CD both are null. Again, this is the scenarios which we have already discussed, like the screen failure and the not assigned scenarios. The fourth is that when the ARM NRS variable is not required if ARM code and actual ARM code are populated. These are the records you can see from the data as the row number 1 and row number 2, where we have both of these information populated. So in this case, since the subject is going as planned, we do not have anything to add to this information. So we are keeping the ARM NRS variables as missing. However, if there is a case that the ARM NRS is populated as unplanned treatment, that is the case where the subject was assigned to a treatment which was not planned, and in that scenario, you will have the ARM NRS variable populated as unplanned treatment, and the actual ACT ARM UD will be populated with the description why the unplanned treatment was given to the subject. The reasons can be such as like by mistake or similar to that. Till now, we have discussed the population of the ARM and ARM related variables for a study where the subject is assigned to single study drug during the study. But what will happen if the subject is assigned to multiple study drugs during the study, and these drugs are assigned in multiple stages? To explain it in better way is, let's visualize a study where the subject is assigned to one drug, and then after the completion of exposure to first drug, the study is unblinded and we have an open label drug being given to each and every subject, irrespective whether it was assigned to which arm it was assigned initially. That is one scenario where the subject is assigned to more than one study drug in this study. Another scenario is when the study is enrolled to a crossover studies, where the subject will go through more than one drug, one after the other during the study. And in these studies, if the subject happens to drop out from the study in between the exposure to the study, so what impact this scenario will have in case for these variables, that is the arm related variables. The best practice for these type of scenarios is that we should create the arm code values as a combination of code representing each stage. For partially assigned subjects, that is the subjects who has not gone through the complete exposures of the different drugs, use truncated codes indicating the completed stage. The truncated actual arm CD value should match with the logic of the arm code. For the arm and actual arm description, we should use the truncated descriptions corresponding to their truncated code. We should ensure that a one-to-one relationship is maintained between codes and their descriptions for all the affected subjects. Another important thing to note here is that the truncated values are not found in TA domain, but they are acceptable only in the multi-stage trials. And these are applicable only to subjects who do not complete all the assignment stages. Let's understand this with an example of a data. Right now, I am showing you a data of 3 subjects. The first row, the subjects 001 was randomized to drug A. At the end of the double-blind treatment epoch, they were assigned to open label A. Thus, their ARM code is AA. 1A for the first drug, that was the drug A, and the second A is for the open label A. They received the treatment to which they were assigned, so the actual ARM code is also AA. For the second subject, the subject 002, that subject was randomized to drug A. However, they were lost to follow-up during the double-blind treatment epoch, so they never reached the open-label epoch, when they would have where they would have been assigned to either the open-label drug A or the rescue element. So in this case, their ARM code is only A because they did not underwent the open-label drug A or the rescue element A. So in this case, this This example illustrates the exception to the rule that ARM CD, ARM, actual ARM code, and the ARM code must be populated with the values from the TA dataset. You can see that we are not populating the AA or the AR ARM code for these values, which are the planned arms for this study and are the values which you will find in your TA dataset. So the second row is also an example to show that the importance that if we create the ARM code values as a combination of code representing each stage, it is easier to populate the values in these variables when the subject has not gone through each treatment epoch but is partially assigned to the treatments. For the row 3, the subject 3 was randomized to drug A but received the drug B. At the end of the double-blind treatment epoch, they were assigned to rescue treatment. The ARM code shows the result of their assignments, that is AR. That is, they were planned to get the AR ARM code, that is the drug A and then the rescue treatment. However, since this subject received the drug B instead of drug A, the actual ARM code shows their actual treatment, that is the BR. Let's talk about the difference between the reference start date and the reference RFX SDTC. Again, here, this is one of the most important concept of the DM domain, which is widely asked in the interview questions, that what is the difference between the RF SDTC and RFX SDTC. So let's understand this concept with with the help of an example. Here is a dummy scenario with corresponding DM dataset that reflects the distinction between the RF SDTC and the RFX SDTC, including a situation involving a washout and a pre-randomization biopsy. The scenario which we are considering here is the interventional oncology study with pre-randomization biopsy and washout period. We are talking only about one subject in this study. Let's say the subject is 001. Now let's understand the data which we have received so far. This subject signed the informed consent date on 1st of May 2024, and it undergoes a biopsy during screening on 4th of May 2024. After that, it enters a 14-day washout period where no cancer medication were allowed. It got randomized on 15th of May 2024, but received the first dose of study drug on 16th of May 2024. It received the last dose on the 8th of August 2024, and the final follow-up was done on 25th of August 2024. So based on the information we have collected so far, let's understand which information will go in which DM variable. This is the information which I am showing you regarding all the date variables which will be populated in the DM domain. And now let's go and understand how these are populated. We know that the subject signed the informed consent on the 1st of May 2024. So it was easy, and we can say that this date becomes the informed consent date, that is the RFIC DTC. The next is that is the date of where the subject undergoes biopsy during screening, and that is the date of 4th of May 2024. So here this date becomes as the RFSC DTC. Why? Why this date? Because we have not exposed this study, we have not given the subject any study drug. So why the date of biopsy is considered as the RFSC DTC? It's an important thing because RFSDTC is not the date of first. It's not the date of the first study drug, but it is the date which marks the start of the trial for the subject. The marking of the start of this trial for any subject can be, and in most of the scenario, is the exposure to the first dose of study drug. That is the reason there is a confusion that RFSDTC is the date of the first dose of study drug, but that is not the complete definition. RFSDTC is the start of the trial for a subject, and the start of the trial can be uh, can be the date when the subject has enrolled to the study, or when the subject has signed the informed consent date, or in this example, if you see, this is an oncology study, so there was some test or assessments being done during the screening period, which is considered as this start of the trial for the study. So that is the reason that since this subject went biopsy during screening on 4th of May 2024, which marks the start of the trial for the subject, this particular date is populated as the RFSDTC date. Similarly, the RFENDTC date is the date where which represents the last known study activity date. So that's the reason the final follow-up date, which is the 25th of August 2024, is considered as the RFENDTC. Again, please note that the RF SDTC and RF ENDTC dates are the dates which are defined as the uh, dates which gives the start of the study trial and end of the study trial. The start can be, and in most scenarios is considered as the first dose of study drug day, but it can be a different date as different day as well, explained in this example. The next is the RFX STDTC. RFX STDTC is the date which is always the first actual study exposure date, that is the first dose of drug. So since the subject received the first dose of study drug on 16th of May 2024, this becomes the RFX STDTC. And the last dose was given on 10th of August 2024, that becomes the RFX ENDTC. To give further details on these four variables, the RFX STDTC and RF ENDTC variables are the sponsor defined variables. RF STDTC is used to calculate the study day variables. It is usually defined as the date and time when the subject was first randomized or exposed to study drug. This definition applies for most interventional studies when the start of treatment is the natural and preferred starting point for the study day variable, and thus the logical value for RFSTDTC. In such studies, when data are submitted for subjects who are ineligible for treatment, such as the screen failure subjects, subjects who are enrolled but not assigned to an arm, or subjects who were randomized but not treated, the RF STDTC will be null. However, there are certain study designs where the start of the reference period is defined differently, such as studies that have washout period before randomization or have a medical procedure required during screening. As in this example, a biopsy was done. In these cases, the RF STTTC may be the enrollment date, which is prior to the first dose. Because study day values are calculated using RF STTTC, it is in this case the study day would not be based on the date of first dose. The RFX STDT and the RFX ENDTC variables define a consistent reference period for all interventional studies and is not open to customization. These dates will always represent the date and time of first and last study exposure. The study reference period often duplicates the reference period defined in the RF STDTC and RF ENDTC, but not always. Therefore, this pair of variables is important, and they guarantee that a reviewer will always be able to reference the first and last study exposure reference period. The RFX STDTC should be the same as SC STDTC for the first treatment element described in the SC dataset. So in simple terms, if you want to answer the question that what is the difference between the RF STDTC and the RFX STDTC, we can say that RF STDTC is the date of the start of the trial. The start of the trial can be the first dose of the study drug, or it can be the date the subject has enrolled to the study or signed the informed consent form or has undergone an assessment during the screening period, as defined in our study protocol. However, the RFX STTTC will always be the first dose of study drug received by the subject. That's all for today. I hope this video helps you understand the DM domain in SDTM. It's one of the fundamental piece in the clinical data programming. If you find this video helpful, don't forget to like, subscribe, and click the bell icon to stay updated on more videos about SDTM domain, CDISC standard, and SAS programming. Drop your questions in the comments and let me know in comments if you want me to cover any specific topic. Thanks for watching.